Okay, so by now you should have watched the lecture video part 1 where we kind of introduced the concepts of photosynthesis. There is that video that is in its own little lecture video, that little McGraw-Hill video. And now we're going to go through the actual process of photosynthesis. So we talked about capturing energy from sunlight. And we are going to capture that energy in photosystems. In our photosystems, we have light harvesting complexes. These are these molecules that are going to harvest light. They're going to absorb the light that comes into it. And they're just going to bounce, 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 plinky, plink along the energy. Then they're going to send that energy to a primary reaction center, primary electron acceptor. And that primary electron acceptor is here in a reaction center complex. That is going to take an excited electron from chlorophyll A. Where is my pointer? So chlorophyll A is going to have an excited electron that sends up to this reaction center complex. So the sun has powered that whole thing, and now we have an excited electron. That excited electron could go two different flows. One is cyclic and one is linear. Linear is going to go through two photosystems, make ATP and NADPH. Cyclic electron flow is only going to go through photosystem 1. It will make ATP and not NADPH. So linear electron flow is the primary pathway. It makes both ATP and NADPH. But cyclic electron flow can be used if a cell needs a little bit more ATP. So it's building up NADPH more than it's building up ATP. It needs some more ATP, so it can go through cyclic electron flow. That's especially helpful when we're talking about the Calvin cycle, which has a high need for ATP. So photosynthesis happened in the light reactions, the photo part, and then the Calvin cycle is going to be the synthesis part. So the light reactions, getting energy from the sun to make ATP and NADPH. And the Calvin cycle is taking carbon dioxide and making a sugar molecule. In the light reactions, we're going to take that sunlight energy and use that to split water to make those three products. Release oxygen. So we've made our oxygen. We don't need it around. We're going to just let it go right out of the stomata away from the plant. We're going to reduce NADP plus into NADPH. So it's being reduced, it's picking up electrons. This is another electron acceptor. In cellular respiration, we use NAD plus and FADH2 to carry those electrons. Here we're using NADP, NADPH to carry electrons. So this is going to act the exact same way, carry those electrons to get energy to power the Calvin cycle. In light reactions, we're also going to take ADP and phosphate and make ATP, which is also going to be used to power the Calvin cycle and to power other things that your cell needs to do. The Calvin cycle happens in the stroma, so that's the liquid part of the chloroplast, and this is going to be taking carbon dioxide with its one carbon and fixing your carbon dioxide into an organic molecule. I'm sorry, I just lost my train of thought. So it's going to take carbon dioxide along with energy from ATP and NADPH, and 6 carbon dioxides are going to turn into one molecule of sugar. So glucose has 6 carbons in it. Those 6 carbons came from running this Calvin cycle 6 times to turn 6 carbon dioxides into that 6 carbon sugar. So, in the light reactions, we have these two photosystems, and they run backwards from what their names would suggest. So we start with photosystem 2. In photosystem 2, we're going to split water and make ATP. There are 2 molecules of P680, which is one of the chlorophyll molecules, and it's going to be excited by light and lose electrons. 2 molecules of chlorophyll means 2 electrons are going to be excited. So they're each going to excite 1. Then that light energy is going to split the water, And those electrons from water are going to replace what P680 just gave up, but it just made excited. When we split the water, we're going to produce oxygen and give it off. And then hydrogen is going to be pumped across a concentration gradient. So we are preparing an electron transport chain. We are pumping protons, creating this proton motive force, the same way that we saw in cellular respiration. So from the energy of sunlight, now we are pumping protons across the cell membrane, creating a concentration gradient where a lot of protons are on one side of the membrane and not as many protons on the other side of the membrane. Those protons are not going to like having that gradient. They're going to create a proton motive force. So the protons are going to move through ATP synthase, and that will produce ATP by those protons going through chemiosmosing through ATP synthase. Those electrons that were lost by the P680 molecules are going to travel on down the cytochromes. And this way of generating ATP through this photosystem, that is called photophosphorylation. Photophosphorylation means that you are creating ATP, phosphorylating ATP through photosynthesis. Okay, now after photosystem 2, we're running through photosystem 1. Photosystem 1 produces NADPH. So the electrons from photosystem 2 are going to travel over here to photosystem 1. And they're going to allow P700 to give up electrons and be excited. Those electrons are going to be excited. So they come in and replace those electrons that are excited. And those excited electrons are going to travel on down. We're not going to worry about these enzyme names, but they're going to travel on to NADP plus reductase, and that is going to take NADP and turn it into NADPH. So NADPH is now carrying a lot of excited electrons. Those electrons in NADPH came from photosystem 1. So what we saw was a photon of light excited an electron in photosystem 2. And that excited electron, the energy was used to create a proton motive force, spin around ATP synthase, make ATP. That electron travels to photosystem 1, where another photon of light will excite it again, and that electron will be transferred to NADPH. So now NADPH is carrying this electron. That electron is excited. It has a lot of energy. And that excited electron, the energy from that excited electron, is going to be used to make sugar with a lot of energy. So we saw ATP being made by chemiosmosis in both chloroplasts and mitochondria using different sources of energy. So mitochondria were taking chemical energy from food to make ATP, and chloroplasts are taking light energy from the sun to make chemical energy of ATP. So light reactions make ATP and can increase the potential energy of electrons by moving them from water to NADPH. So if you should at this point be able to kind of follow me along as I'm going through this picture. And if you don't, kind of make a note to come back and make sure that you can understand what's going on in this picture. So in photosynthesis, so far, we've only done the photo part. We've taken a bunch of energy and converted it into ATP and NADPH. So what happened, a photon of light, a little piece of light shown on photosystem 2. It went through the light reaction. It went through this excited an electron. That excited electron moves to this moves to this electron transport area that is going to create a proton motive force, pumping hydrogens into this space. Those hydrogens are going to then go, as they accumulate within the thylakoid space, you're getting a high hydrogen concentration, high proton concentration. They're going to want to go back to equilibrium with what's outside the cell, or what's outside the thylakoid. So they're going to go through ATP synthase. Their movement through ATP synthase triggers ATP to spin around and photophosphorylate ADP to ATP. So we've made ATP. Back here with our electron, it keeps on traveling through these membrane complexes to photosystem 2. When it gets to photosystem 2, another photon of light will excite this photosystem to, another photon of light is absorbed by this photosystem, by photosystem 1, and that causes the electron to be, I just lost my thing, that causes the electron to be excited and carry a lot of energy. The energy in that excited electron moves through NADP plus reductase and gets carried by NADPH. Okay, so NADPH is carrying energy in the form of excited electrons, and ATP is carrying energy as the energy currency of the cell. And those are both going to go to the Calvin cycle, and they're going to provide the power of the Calvin cycle to take carbon dioxide and turn it into sugar. So carbon enters the cycle as carbon dioxide, CO2, and it is going to run 3 times, 1 carbon dioxide, 2 carbon dioxide, 3 carbon dioxides. Now you have a 3-carbon molecule called G3P. G3P's full name is glyceraldehyde 3-phosphate. That's a bit of a mouthful, so we're going to call it G3P. Then we're going to take 3 more carbon dioxides, 1, 2, 3, and make another G3P. 2 G3Ps hook together and make a 6-carbon sugar, and that gives us glucose. So to make 1 G3P, the cycle runs 3 times because you got to stick in 3 carbon dioxides. To make an entire glucose, you're going to have to make 2 G3Ps, stick them together to make glucose. This cycle is anabolic. It requires energy, and the energy is provided by ATP and NADPH. ATP and NADPH are, helpfully, the 2 things you just made in the light reactions. The carbon cycle has 3 phases. First, we fix carbon dioxide. That means we are going to take carbon from carbon dioxide and turn it into not carbon dioxide. We're going to turn it into a bigger organic molecule. Then we have reduction. So NADPH gives up its excited electrons. Those electrons are going to be incorporated into G3P. So G3P is holding a lot of energy in its electrons now. And then we have regeneration of the carbon dioxide acceptor, RUBP. So it has to come back up here to be able to have this whole cycle happen again. An enzyme to pay attention to here is Rubisco. That's an important enzyme. We'll talk about it a little bit more. Rubisco is what takes carbon dioxide and grabs it out of the atmosphere and brings it in here. So your cell has brought carbon dioxide through its stomata into the I just forgot my word. Oh my goodness. Your cell has brought carbon dioxide through the stomata into the chloroplast. Rubisco grabs that carbon dioxide to fix it into here. So in the light reactions, these are going to be carried out by molecules in thylakoid membranes, whereas the Calvin cycle is taking place in the stroma. That's 2 facts you need to remember. Light reactions convert light energy from chemical energy from food or glucose to ATP and NADPH. And Calvin cycles take that ATP and NADPH and use it to convert CO2 to G3P. G3P is a basic sugar building block that is going to be used to make glucose. In the light reactions, we split water and release oxygen to the atmosphere. In the Calvin cycles, we return ADP, inorganic phosphate, and NADP plus to the light reactions. So that's kind of all important stuff that you need to be very comfortable being able to tell me each of those happen, where it happens, what is happening. So light shines on your plant cell, and those chloroplasts are going to pick up that light through photosystem 2. Photosystem 2 is going to take that photon of light and use it to excite an electron. The electron travels through an electron transport chain to make ATP. that excited electron can travel on to photosystem 1, where another photon of light can where photosystem 1 will use another photon of light to excite the electron and create NADPH. ATP carrying energy and NADPH carrying excited electrons to the Calvin cycle to power the Calvin cycle. As a result of this, oxygen is breathed out, or oxygen is released as a waste product. So ATP and NADPH come to power the Calvin cycle. Carbon dioxide from the environment is brought in to the stroma through the stomata of the cell, and 3 carbon dioxides are used to run the Calvin cycle 3 times to make G3P. G3P is a building block for sugar. Sugars can be used to do cellular respiration, whether it is the plant itself that needs to go through cellular respiration, or it is something that has eaten that plant and is going to turn that sugar into energy through cellular respiration. The sugar can be used to build cellulose, which is a structural polysaccharide in plants, makes its cell wall, helps keep those cells nice and firm, and that sugar can be turned into starch. Starch is the storage polysaccharide in plants. It allows them to keep some extra energy stored for when they need some energy. And that sugar can also be used to make other organic compounds. So if that plant needs to make proteins, or it needs to make lipids, or it needs to make nucleotides, all of those can be made from that basic sugar. Okay, let's talk about a few evolutionary adaptations for some plants. So in some plants, having those stomata open all the time to let oxygen go out and carbon dioxide come in is a little bit of a problem, because it leaves the plants dehydrated, and plants don't want to be dehydrated. So on hot, dry days, they're going to close their stomata to hold that water inside of them. But as much as that keeps the water in the plant, it's going to limit photosynthesis, because carbon dioxide can't get in, and without carbon dioxide, you're not synthesizing anything. It also causes oxygen to build up. So oxygen is a waste product here. It's not going to help make photosynthesis run. It needs to just get out of there. If you're building up oxygen, it's just going to get in the way of photosynthesis. And so without carbon dioxide and with too much oxygen, suddenly Rubisco doesn't have access to the carbon dioxide to run the Calvin cycle, but it's got oxygen all over the place, and sometimes it's going to end up grabbing oxygen and wasting energy running the Calvin cycle with oxygen instead of running the Calvin cycle with carbon dioxide. That's a pretty energetically wasteful process. So Rubisco is supposed to incorporate carbon dioxide in the Calvin cycle. Rubisco is so important. It's about 30% of the soluble protein in most plants, but it is not really good at discerning between if it is picking up carbon dioxide or oxygen. So plants need to get that oxygen out of the way, and they need to get the carbon in there so that Rubisco is surrounded by carbon dioxide. Okay, excuse me. Okay, so the plant needs to get lots of carbon dioxide into your chloroplast and needs to get rid of oxygen so that it's not going through photorespiration and wasting energy to run the Calvin cycle without making sugar. So just closing your stomata all the time isn't a great way to run photosynthesis. So what we've talked about so far in photosynthesis are C3 plants. These are plants that take carbon dioxide and form a 3-carbon compound, that G3P. In photorespiration, Rubisco adds an oxygen instead of taking in 3 carbon dioxides, so you end up having to run the Calvin cycle extra times to get that 3-carbon plant. There are some plants called C4 plants, and they're called C4 because they use 4 carbons. They're going to run the Calvin cycle 4 times to make a 4-carbon compound. And these C4 plants that are doing that are making these 4-carbon sugars, have a way of reducing photorespiration. So the way, okay, well, let's throw in this note first. So 85% of plant species on the planet are C3 plants: rice, wheat, soybeans, and all trees. But C4 plants, okay, C4 plants want to minimize photorespiration so that they can keep their stomata closed and conserve water. So C4 plants have developed this way to physically separate light-dependent reactions and the Calvin cycle. So the light-dependent reactions are going to happen in mesophyll cells. So that oxygen that is being created is going to be held in the mesophyll cells and won't be able to get to rubisco. The Calvin cycle then is going to happen in the bundle sheath cells. So rubisco has no access to oxygen. The Calvin cycle is happening in the bundle sheath cells, and it'll only have access to these carbon dioxides. To the carbon dioxide that have been brought in when the stomata were open. So the Calvin cycle runs and makes a sugar. Some plants are called CAM plants. They use a CAM pathway. And CAM pathway is another way to minimize photorespiration. CAM stands for crassulacean acid metabolism, which is another mouthful, so we say CAM plants. CAM plants include cacti and pineapples. Instead of separating light-dependent reactions and Calvin cycles in different parts of the plant, they're going to separate them in time. So the photo part, the taking energy from light, has to happen when the sun is out. So it happens in the daytime. But at night, so that photo part capturing the sunlight, they're going to have their stomata closed so that they're not being dehydrated. At night, the CAM plants are going to open their stomata. When they open their stomata, all the oxygen that built up running the light reactions, all that oxygen can be released. All the carbon dioxide that's out in the air can be brought into the plant. And then at nighttime, with their stomata open, is when they're going to run the Calvin cycle. In CAM plants, carbon dioxide is going to be fixed into oxaloacetate, and then it's going to be converted into an organic acid. That organic acid is going to be held in the plant until the next day, and in the daylight, they are going to keep their stomata closed, but they'll run the photo part, the light reaction again. When the sunlight is out, when the stomata are closed, those organic acids are going to be broken down to release carbon dioxide to enter this Calvin cycle. And this controlled release will keep a high concentration of carbon dioxide around rubisco. So we are not running the Calvin cycle with oxygen. We're only running the Calvin cycle with carbon dioxide. Okay, so C4 plants separate the light reactions and the Calvin cycle, or the dark reactions, in different types of cells, mesophyll cells and bundle sheath cells. In our CAM plants, they're separated by night and day. Okay, all life on our planet depends on photosynthesis. So energy enters chloroplast as sunlight. That is going to be stored as chemical energy in organic compounds in the plants. The sugar made in the chloroplast will supply chemical energy. It also supplies carbon skeletons to synthesize organic molecules of cells. Plants store excess sugar as starch in their chloroplasts and in their roots, tubers, seeds, and fruits, those delicious parts of plants that then all of consumers are able to come eat and grow and have energy to do all the things that consumers need to do. Okay, so please make sure you're comfortable with all of that photosynthesis part, because we will have a photosynthesis quiz when we are back in class. And I will see you all later. Bye.